Welcome to All the Best, an interview series with UNL Orca awardees, where UNL means University of Nebraska Lincoln, and Orca is the Outstanding Research and Creativities Award, which is UNL's top award. In Gerda Lerner's book, Why History Matters, she explains that life stories give meaning to the present and the future because we identify what is important to us. And she says, our stories show how we are interconnected in a particular time and place. As for this interview series, Orca awardees have experiences that reach back more than a half a century, which will give us a personal view of how the best research emerged from UNL. It is my pleasure to introduce today's interviewee, Dr. James Van Etten. He was awarded the Orca in 1992. After I provide a quick summary of his life and career, Jim will tell us about his time at UNL, including his award-winning research and about the rest of his life. Jim was born in Virginia, but grew up in Peoria, Illinois. Something in his youth must have sparked an interest in science because he earned a bachelor's degree in biology from Carleton College in 1960. Carleton is a well-regarded liberal arts institution in Northfield, Minnesota, about 400 miles from Peoria, so he had to go quite a distance from home. Next, Jim earned a master's and doctorate at the University of Illinois in Champaign-Urbana, both in plant pathology. For his postdoctoral work in 1965 and 66, he worked in the Department of Genetics at the University of Pavia in Italy. Immediately after the postdoc ended, he joined the University of Nebraska Lincoln as an assistant professor of plant pathology. As we'll learn, he's had a remarkable research success as he rose through the ranks, so much so that he was elected to the National Academy of Sciences in 2003. With that, I want to welcome Jim to the interview.
Thank you, Mark.
First question is about your first impressions of the campus when you saw it for the very first time versus what you think about it today.
Okay, that's an interesting question because when I almost didn't even come out for the interview, a week or two before I got a call from Nebraska to come for an interview, I had just received a National Science Foundation fellowship to go to Italy, and but it turns out that I talked to a couple faculty at Illinois, and they said, well, Nebraska has a couple very good people in the department. You should come out and take a look at the place. So I came out, and it was actually in the middle of March, and it was a snowstorm. So the plane, at that time, the plane from Chicago stopped at Omaha and then flew on to Lincoln. So we were about 4 hours late getting in. It was around midnight into Lincoln. And the chairman of the plant pathology department by the name of Mike Busalis picked me up at the airport. But he said, I've got to wait a minute because I've got to pick up 3 people who are coming from the National Science Foundation to look at the chemistry department at Nebraska about funding their new building. And so anyhow, the 4 of us piled into Mike's car, and we were driving probably somewhere on Holdridge Street. And one of the fellows from the NSF said to Mike, what's it like living in Lincoln, Nebraska? And Mike said, oh, and we were at a stop sign, and Mike said, it's very laid back. Well, anyhow, the light changed, and the car behind us just laid on the horn, and then it pulled around us, and it had a New York license plate. So that was one of these stories that really happened, and I'll never forget it. And obviously, the NSF people liked what they saw because Nebraska then ended up building Hamilton Hall, which is a big building on the center of the UNL campus. Well, anyhow, so I say my time here for those 2 or 3 days was essentially during a blizzard late spring, because everybody I met was telling me, gee, you should have been here last week when it was 70 degrees and beautiful. But anyhow, in terms of the overall campus, it's totally changed. East Campus, my first lab was actually in what is now called entomology building. At that time, it was the plant science building, and I had the top floor. The plant path department was located in the top floor, and so that's where my lab was eventually going to be. But the East Campus Union now sits on the parking lot where we used to park for that building. And of course, since then, there's been all kinds of new buildings on East Campus. The city campus has totally changed too. When I was first here, the center of the campus was a parking lot, which now has the library extension plus lots of statues and so forth. And it's very impressive. But that time it was just really a parking lot. And 14th Street actually ran through the campus, which is now blocked off. So there's been a number of changes in that time and many new buildings, lots of new excitement going on. So yeah, it's been interesting in the 60 years I've been here to see all these remarkable changes.
Great. So let's jump right into your award-winning research and give us a sense of how that came about, and then what it actually is, and why is it important?
Okay, well, I've actually worked in 3 areas since I've been at Nebraska. I was actually hired to work on the biochemistry of fungi. As Mark said, my PhD degree was in the plant pathology department at the University of Illinois, but I actually worked in an antibiotics lab because that happened to be the lab where the department my advisor was located in. And so I was actually hired in Nebraska to work on the biochemistry of fungi, which I had done when I was a grad student. And when I interviewed him, I told him I would probably work on mode of action of fungicides because that was pretty much a wide open area. Then after spending a year in Europe, I was very fortunate to be in a lab that was involved in the early days of molecular biology. And I just fell into a project that they were working on, which involved looking at in vitro protein synthesis, which in those days consisted of using polyuridylic acid, a messenger RNA, making polyphenolalanine. And what we discovered was that we made this system from 2 bacteria and 3, now we would call eukaryotic organisms. At that time, that term didn't even exist. We had a system from a plant. I made a system from a fungus, and we had a system from an animal cell biology, grown cell biology. And we discovered that if you isolated ribosomes from the 2 bacteria, they would work with enzymes from bacteria to make protein. And the 3 systems from plant, fungi, and animal cells those ribosomes would work with the enzymes to make proteins, but you could not exchange these with the 2 bacteria. And so we had a paper in Nature and one in Journal of Molecular Biology. And very shortly thereafter, it was discovered that ribosomes, or that bacteria, the size of ribosomes from bacteria were different than those from plants, animals, and fungi. And so this work was very quickly made sense and was actually reviewed in Nature at the time we published it. So I then came to Nebraska thinking that I'm going to work on in vitro protein synthesis in fungi, specifically looking at fungal spores. And when you have a spore, it really sits there and does not have much metabolism, if any. And all at once, when you put it under the right conditions, 2 or 3 hours later, it's making protein, RNA, and DNA very rapidly. So I was interested in the question of do ribosomes and the enzymes transfer RNAs and so forth, do they work in the spore? And what was the difference between them and germinating spores in terms of these components? Another question we asked, does the spore contain preformed messenger RNA? Anyhow, that was what I started working on, and we published a number of papers on this in the mid 60s to late 70s, and various aspects of this. Now, the second, so that's one area that of the three that I worked on in Nebraska. The second area was that during the year I was in Italy, a lady came to the department who had her PhD from the University of Indiana in microbiology and had come to Nebraska because her husband, Dr. George Vitever, had taken a job in the chemistry department, and his wife was just looking for a place to work. And she was interested in looking at viruses that infected bacteria, where the bacteria infected plants, with the idea of looking for biological control agents, things. Well, anyhow, she came to the plant path department, and they said, "Oh, we'd be glad to have you work in our department. And by the way, we have this empty lab sitting here," which was going to be my lab. So anyhow, for a number of years, Ann and I shared the lab. But the second thing that I started working on was, it turns out that Ann discovered a very interesting bacterial virus, which she thought at the time might have a lipid envelope on it. Now, in the meantime, when I took the job, I was told that there was a lot of money that had to be spent in the next couple months before I was even finishing my PhD, or they were going to lose it. So I bought some equipment, including a gas chromatograph, which I'd used at Illinois. And one day, Ann, then back in Lincoln, said to me, "Do you know how to make that thing work?" I hadn't even taken it out of the box because doing molecular biology, I didn't need that. I said, "Sure, I think I can make it work." And set it up, and sure enough, we found that the virus that she had isolated had fatty acids associated with it, nuts. And at the time, it was only the second bacterial virus that had lipids associated with it. Furthermore, ours had an external lipid membrane, which was the first bacterial virus that had that property. Now, the second thing that we learned was we asked the question, what kind of nucleic acid did this virus have? And the other person that has to be mentioned in this story is Myron Brackey, who was a USDA plant virology scientist assigned to our department. By the way, Myron Brackey was the first person in the state of Nebraska to be elected to the National Academy of Sciences. Well, anyhow, I asked him, how could we determine what kind of nucleic acid the virus had? And he designed the perfect experiment, and it was using sucrose density gradients, which was a technique that Myron Brackey had developed a few years before, and what was led to his election to the National Academy of Sciences. Well, anyhow, I I did what Meyer told me to do. I was scanning the gradients, which to look at the nucleic acid, and he knew exactly what we had, but he told me it over 3 days. First, he says, "That looks like a double-stranded RNA based on your experiments." And I said, "Oh, okay." And the next day he said, "By the way, there are no known bacterial viruses with double-stranded RNA. And the next day he said, by the way, there's 3 components to that double-stranded RNA. And of course, there's no other bacterial viruses with split components. Oh, that's interesting. Next day came in and said, double-stranded RNA can maybe induce interferon. Now that I did know a little bit about because I'd interviewed at a couple of drug companies and had job offers from them. And I knew that Eli Lilly pharmaceutical company was very interested in looking for interferon, working with interferon, which at the time was considered an antiviral mechanism of some type. So I immediately called Eli Lilly to my friends at Eli Lilly who had taken jobs there, if they thought Eli Lilly would be interested in the virus. And 30 minutes later, they called back and said, yes, we're very interested in the virus. Hire a patent lawyer, we'll pay for the patent, and we'll be glad to work with you on its ability to induce interferon. Well, at that time, back to what Nebraska was like at the time, they had no facilities in terms of getting us a patent lawyer. In fact, after a year, Ann Vitterer, who had discovered the virus, finally hired a lawyer to force the university to get us a patent lawyer, which was not even going to cost them a penny. So anyhow, we eventually, and we couldn't publish on it during this time because we didn't, at least in those days, and I think it may still be true, if you patent something, if you want to patent something, you have to patent it within the time, the first year after you publish it. Okay. Mark's shaking his head. Yep. Yes, I gather that's still the case. Well, anyhow, the word had leaked out, and I remember even getting phone calls, and somebody from the University of Wisconsin Medical School was going to sue us if we didn't give them the virus and so forth. But anyhow, we eventually got a patent, which made exactly zero money. Like most patents, I have 2 or 3 others that are in the same boat. And we published it. And so that was the second area I worked on. We did a lot of things with that phage or bacterial virus, along with working on the spore germination.
What was the name of the phage?
It's 5/6 is the name of the virus. Okay. And that virus has got a long history. There's 2 people who made careers studying 5/6, and in recent years there's been quite a bit of interest in using it to study evolution history of bacterial virus, and so it's still a well-known virus among the bacterial virus people. So now back to the third area that I worked in. And this all started in probably about 1979 at my house. I was hosting a visitor or seminar speaker or job candidate or somebody. That part I don't remember, but I remember talking to the former director of the School of Biological Sciences, Russ Meitz, in my kitchen, and we were I was talking to him about this bacteriophage, and he says, my alga has a virus. Ooh. And I said, what are you talking about? And for some reason, I knew there was essentially nothing known about viruses infecting algae, eukaryotic algae. And so he said, I don't know. He said, I had a visitor in my lab the other day, and he looked through a notebook I had and said, gee, your alga looks like it has a virus. Well, anyhow, I stopped down in his lab a couple of days later. And first of all, I have to go back and tell another story. So we were drinking beer in the kitchen when this conversation was taking place. And I've given a seminar on these giant viruses that I'm going to tell you about in a minute, probably at a I don't know, 50 or 75 different universities. And so I pretty much know the type of questions I'm going to get. But somebody, I think it was at Northwestern University, said, "What type of beer were you drinking?" And I says, "That I don't remember, but if it was at my house, it had to be cheap beer." So anyhow, back to the virus story. So
An important
that's the only
interjection.
time I've ever gotten that question. That always struck me pretty funny. So anyhow, I stopped at down to Russ Meitz's lab. He's on the city campus. I'm on the east campus at the university. And we looked at his notebook, and Russ told me what type of research he was doing. I didn't know him that well beforehand. And he studied certain organisms like Hydra, Paramecium, and some of these small organisms have algae in them that are symbiotic organisms. And he, at the time, he was studying Hydra, and he was interested in the question of how did the Hydra, when it took up the green algae, how did it know it was the right one to form a symbiont? And others were taken up and just digested by the Hydra. So, but he also said they were unable to grow the algae that were symbionts in Hydra. And he had isolated the algae and every few hours or first 2 or 3 hours had taken pictures, had the electron microscopists take pictures of them. And it literally out of a notebook of about 100 pictures of these algae, there was one that had what looked like virus-like particles in them. And second, he had written on the back of the photo that it had been isolated and set for 3 hours before the photos were taken. So we thought, well, gee, you can't grow the algae. Maybe there's some connection to virus particles. So we then did the most obvious experiment, isolated algae from the Hydra, let them set for various times and sent them to the microscopist to look at. And by 24 hours, all the algae were just totally full of virus and were lysine. So that was our introduction. Now, again, the first EM pictures we had, we went out to Myron Brackey again. He keeps coming up, and Myron would never let us put his name on any of the papers that we publish, both on 5.6 and on these giant viruses I'm talking about. Of course, we acknowledged him in all these, but he immediately looked at him and says, "Man, those viruses look like they're big," and so and the other reason I'd gotten interested in him, at the time, going back to the kitchen story, uh, I knew that that I worked with fungi, and at that time, several labs were discovering that fungi had viruses, and all of them had a double-stranded, or most all, had double-stranded RNA genomes. So I always thought of algae as just green fungi, and there might just be another double-stranded RNA when we first were talking about this project. So anyhow, Myron took a look and said, "These look awfully big." So then I started working with Russ, where he would grow the Hydra, bring them out to my lab. I would isolate the algae and then let them sit for overnight, and then we'd purify virus from them. And we very quickly determined that I was totally wrong on the genome, that these had double-stranded DNA genomes. And at that time, restriction endonucleases were just being commercialized, and we could take a piece of DNA from, or take the genome from these large viruses and isolate the DNA, cut them with a restriction enzyme, run them on gels, which were now available to do. And we discovered you could add up the size of all the fragments and we quickly discovered that these had very large genomes, over 300 kb, which at the time was bigger than any known virus.
So 300,000, and then the
Yeah,
normal ones
300,000.
were 5,000 or smaller.
Well, most of the viruses that you're familiar with like human viruses, like COVID and flu and HIV, they have 10 or 12 genomes and probably have genome sizes of about 10,000, 15,000 bases.
Okay.
Now, the smallest ones, there are some as small as 5,000. But at that time, the biggest one known was the relatives of the smallpox virus, and they had genomes about half the size of ours at that time. And so that was the first of many exciting things. Now, what really made the project work was, see, the only way we could grow the virus was, in the first few years we worked on it, was we'd get the hydra, isolate the algae, and then let the virus develop in which were already infected, develop. So that was fine. We had a couple nice papers, but to really work with the virus, that was not going to go very far. So anyhow, the undergraduate that used to bring the samples that Russ grew out to me to isolate the algae from hydra said to me one day, I've been reading about Paramecium bursaria, and they have algae in them. What if I grow some and bring them out? Would you see if they have virus? And we did the experiment, and sure enough, they had viruses too. And we knew the viruses were different from one another because we could look at the digestion of the genomes with restriction enzymes, and the fragments were different. So,
but still 300,000.
Oh, these are all big, yeah. No, the 300,000 to 350 or so. And so we then at that point, there had been reports of people who had claimed they had grown the green algae from hydra independently of the hydra, and also of paramecium independently of the paramecium. So in those days, this was before email, I wrote probably 8 or 10 labs that claimed this, and we got cultures back from most of them, and several of them were so badly contaminated you couldn't do anything with them. But I got 5 or 6 of them to grow, and then tried to infect them with the various viruses that we had isolated. And it turns out that the viruses that came from the algae from paramecium that this undergraduate had talked us into working on would actually replicate and in an alga that supposedly had come from paramecium. Furthermore, we found that we could plaque assay that virus, and this was right before Christmas, probably in 1982 or 83. And I think everybody in the building knew that we could, they didn't know what a plaque was or anything, but heard me yelling about, gee, because I knew at that point, then you could really do studies on the virus because you could do it independently of the organism that the alga was growing in. So that was a very exciting day, and we then I spent at some point there over a 2 or 3 year period, I made the transition from dropping the other 2 projects, which had been very successful. We had NIH money supporting them and so forth, to working on these algal viruses. I found out later that some of my colleagues thought I was nuts for doing this. Maybe it was male menopause, I don't know what, but anyhow, I've never regretted making that switch, and we certainly had a very fundamental, successful career for it. And back to, you asked the original question, how to get elected to the National Academy of Sciences. I think it's primarily from the work with the algal viruses because they turned out to be the first of the so-called giant viruses now. And they've done all kinds of interesting and strange things that no other virus did up till that time.
So once you had the plaque assay, is that when you then found that the virus is in all open bodies of water?
Yeah, well, our viruses infect algae that only grow in fresh water.
Oh, okay.
We've not been able to.
Oh.
I mean, it turns out salt water kills the host.
Mm.
Now,
Mm.
subsequently, viruses have been found in ocean water that infect algae that are related to ours that are big and so forth. And some of those you can plaque assay. And so these viruses, like what we have found, we've now found them in fresh water all over the world. Well, the only place I haven't looked is Antarctica, but I found them in Australia, Africa, Asia, and so forth. And we now have four different hosts for these viruses, and the viruses are specific for the hosts they grow in, and all the hosts actually are symbionts that grow in other organisms. So there's some very interesting biology going on there, and some of it we're trying to understand.
Great. Well, there's a lot to talk about there, but I'm going to move on. Tell me about recruiting workers to your lab, graduate students, undergraduates, postdocs. How did you recruit them? Where'd they come from? And then when did it all play into your story here?
So I've probably had around 20 PhD students in my lab over the years, maybe one or two more. I actually have never done a lot of outside recruiting. They seem to show up for various reasons. In fact, one with the person doing the interview, one of Mark Greep's former grad students ended up doing her research in my lab. So, I've been very fortunate. I've had some very good students over the years. In fact, my first graduate student who worked on the fungal work, he arrived in Lincoln about a week before I did in the department and was interested in physiology, which was my fungal biochemistry and physiology background. And so he was a very good student, postdoc at Stanford, and then spent his research career at the University of Minnesota and just retired 2 or 3 years ago. Others I've had have been from a number of different countries. I think I've had 3 from mainland China. In fact, one of them was the first grad student, I think at UNL, that came from mainland China and was a very good graduate student. And then I've had several.
Did he return to China?
What's that?
Did he return to China?
No, all of the ones except for one, and some of these have been postdocs also from China, stayed in the United States. Yeah. In fact, he had an interesting story because when he got his degree, then China was wanting students to go back at that point, and the U.S. was supporting this. This would have been about 1986 or so. And he then went to China or to Canada to do a postdoc, and then eventually came back to the States when that rule was no longer important. In fact, he worked with, you may remember, Fred Wagner had a company called Nebraska.
I do.
And Unanan Shaw, that was his name, actually went to work for that company until it went broke. But so and then he came back to the university and ran the biotech sequencing, DNA sequencing laboratory.
I probably met him.
I
probably have met him.
And was very good at it. So So other grad students, I've had several that have done their work in my lab from foreign countries and then go, but their degrees actually from a foreign country. I remember one lady from Vienna, Austria, went back, another one from Egypt. Trying to think who else now, but that has happened quite a few times.
So very exciting. You and I talked the other day about your most cited papers, and I hope you remember what they are. And the reason I bring this up is because you've got this amazing research projects, 3 of them, and yet the most cited papers that people have often don't correlate with their most exciting research. And so tell us about the most cited papers.
Yeah, I didn't know what it was until Mark sent me an email a couple days or about a week ago. So I guess the most cited one is one published in nucleic acid research, and it was a paper led by Rich Roberts, who's a former Nobel Prize winner, and on the nomenclature for restriction endonucleases. And so one of the issues, and this is, I don't remember what it was published in the 90s, maybe, or somewhere in there. Yeah. Anyhow, people were using different types of names for naming DNA restriction endonucleases when they were isolated, and they were trying to get a uniform policy on naming these. And since Rich discovered some of the early ones, it turns out that's not what he got the Nobel Prize for. He got it for this, being a co-discoverer of introns in RNA, but he discovered a number of the restriction enzymes, and then he worked with New England Biolabs on giving them the enzymes, and essentially after being at Harvard and then at Cold Spring Harbor, actually moved back to New England Biolabs in Beverly, New Hampshire, and finishing out his career there. But so that's apparently been cited several hundred times.
560 times or something? It was a lot.
That's
a very large number.
Yeah.
And your second one was the genome sequence of PBCV2, I think?
I'm trying to remember what the second was now. You told me, and I can't.
Yeah.
It was one of the host algae, the sequence of one of the host algae that the virus infects. And it turns out that was the second alga to be sequenced. The first one was Chlamydomonas, which is a model alga. And I think that's why that was cited so much. And the alga actually came from Antarctica, as I remember the history. But yeah, so we then, when it was sequenced, we had the sequence then, and this again was in probably the late 90s or early 200s or 2000s, and it got a lot of interest just because that's a time when people were sequencing these various eukaryotic organisms and so forth. So, but we had now both the alga and the host sequence, and that allows you to do certain types of experiments that you might not. Yeah.
So the flip side sort of question is, are there things that you've published that you feel are undercited or underappreciated? You recall anything?
Well, certainly, I don't know if I'd call it underappreciated, but ones that I would have thought would have been more highly cited. I mean, the discovery that that we could plaque assay the viruses, that was actually published in Science, and we had hoped to get the cover of Science, which had a nice green lawn with circles on it that were where the virus infected killed the algae. And it turns out there's an artist that determines the cover photo, and she wanted to know what the holes in the green lawn was. But she had just 2 or 3 issues before Science had apparently a Petri plate on the cover, and so she just decided not to put. So the paper was published in Science, but not the cover or not the color picture of a green lawn with the plaques in the back. So that was certainly, and it's been cited quite a bit. Certainly, then some of the discoveries with the virus, these giant viruses, have been phenomenal. I mean, the fact that we discovered that they, and again, back to that Chinese student I mentioned, we discovered that these viruses, which can be found in freshwater essentially all over the world, one of the genes they code for are very interesting, including they encode for these DNA restriction endonucleases, which was the first time that these enzymes have been found outside of bacteria. And the story is backwards because bacteria produce these enzymes to prevent virus infection or foreign DNA getting into the bacterium. And in our case, it's the virus that encodes for the enzymes, and we're dealing with not a bacterium, but a eukaryotic host. So, and some of the enzymes are unique and are actually sold by doing the biolabs. And I think we were talking about patents earlier. We've got some patents on these, and most I did make a few $100s a year on them. I could spend as I tell people, I could spend a weekend in Omaha at a cheap motel on the royalties.
You could buy better beer.
But anyhow, that discovery again. I mean, I'm sure these papers have been cited, but that's one thing I've never done is look at the number of citations. And then one other one was a science paper. We discovered that the viruses encode for the world's smallest potassium ion channel protein. And we worked with two biophysicists in Europe. It's actually not a husband-wife combination. He was in Germany and she's in Italy. And that there's now over 60 publications on the potassium ion channel encoded by these viruses. And so, and again, I say the first paper was in a science paper, and a second science paper on that subject was the group in Europe. They isolated a protein that was sensitive to light, and they hooked it up to our channel, and the channel can open and close in red and blue light. Then they put the channel in zebrafish, and they could change the behavior of the zebrafish by what the color of the light was, like red or blue, in the lab. And they've done many more things with these viruses now, or I mean with these channels now.
No, amazing. So with your research prominence, I'm going to open this up to a more complicated topic. So tell me about editorships, patent applications you've done, companies, books you've written. It's a big topic, all the different things that research scientists do that nobody really fully appreciates.
Well, in terms of editorships, I've been on the editorial board of probably a half dozen journals now. I think right now I'm only on a couple because some of these have a time limit to them. But so you spend a lot of time reviewing manuscripts and commenting on them. I've also been involved in reviewing a number of grants. I've served on NIH study section. Actually, back when on my fungal work was the reason I was on NIH study section. And at the time I was on it was actually when I was making the transition to look at these giant viruses. So, and I've been on the U.S. Department of Agriculture, had grant panels, and I've been on one of those a couple times. So I've served there. What was the rest of your question?
Well, so when you're doing all these reviews, how is that important for you as a scientist? And then why do scientists do this?
Well, that's a good question because you're certainly not compensated for it in any way, and it does take a lot of time. But I think all of us who are practicing scientists want to see good science get published, and that's what we strive for. And to a certain extent, I suspect all of us that do a fair amount of reviewing have that in the back of their mind. But it's not, it does take, there's no question, but it takes a lot of time to do a good review. And I still get papers to review. I turn down more now than I probably review than maybe I used to, just for time and some areas, of course, I don't know anything about them, so I don't review.
So then the second one was patent applications. I know you've written patents, and that takes a lot of work. And I don't know, why would somebody do that? I think the public thinks we do fundamental research and then it has to transition somehow to some useful product. But how does that go about? How do you decide what to patent?
Well, that's a
Or
good,
write a patent application for, sorry.
yeah. So certainly UNL, as I told you about my early experience with patents here, they've been very interested in patenting things, but I think I have 4 patents in total. 3 of them are on various aspects of these giant viruses, and I'm trying to think. One of them I think was written by somebody else, and but they do take time and so forth. And so the reason you do this is to protect intellectual property that might eventually have some commercial use. And in fact, the first restriction endonuclease we discovered, I actually talked to the company that we had worked with, New England Biolabs, about getting a patent. And they said, we don't patent any of ours at the time. Now, subsequently, I think that's changed. But at the time, in the initial days, they actually didn't encourage it, didn't suggest.
I would have assumed you did patent that, but yeah.
No, that was okay. Now, one enzyme with another commercial company, who I think I may be on the patent one of our enzymes, but that company had lots of issues beforehand and went broke a long time ago. So now I'm trying to think. Well, okay, so one of the things the virus could do is when people were first making plants with using recombinant DNA, they wanted to be able to take the DNA of the gene they're interested in, and they would put a sequence before the gene called a promoter, so that the organism would express that particular gene. And it turns out that the promoter elements from these viruses work very well in both plant. Actually, it turns out one of the few that work both very well in plants and bacteria. And there were a couple patents on that. I think I was on one of them. A couple of them I was not even on the patent application itself. But we were probably just a little late in discovering that, because the plant people had discovered that another element would work very well as a promoter in a lot of higher plants, even though ours probably even worked a little bit better. So that was never used very much. But yeah, the patent process is interesting. As now, a lot of universities, like the University of Wisconsin, have made a heck of a lot of money off of patents. Yeah. You know what I'm talking about.
So then this continues. Did you ever start a company or co-start a company, edit books, that sort of thing?
Okay. Well, I actually considered starting a company when we discovered these viruses made restriction enzymes, and different viruses made different restriction enzymes. And I gave it some thought, But, and then we also had a person who came to Nebraska who had been at New England Biolabs and had a company here. And we talked about him commercializing our enzymes. There were issues and that never worked out. But the best thing I ever did was not start a company on the enzymes. I mean, after we discovered that these viruses encoded 5 or 6 enzymes that we purified, we got bored doing that. So there was too many more exciting things to find out about the viruses. So in terms of editing a book, I edited one book on the unusual giant viruses, I don't know, 20 years ago. And we talked about some other viruses that were at that time just being discovered, some of which were bigger than ours. So, yeah, that was a fun experience. And again, I don't know how many copies of the book they sold, hopefully more than the toes on my feet.
Right. You got to wonder who's buying these books. So tell us about the organizations that funded your research, and then how did they change over time in your relationship with the funding agencies?
Okay. So this is interesting in a way. So when I came to Nebraska, I only found out that the dean of the ag school at the time didn't want faculty applying for grants because they lost control over them. I didn't know any of this. So shortly after I got here, I wrote a grant proposal to NIH, and I don't know how it got out of the university, but it got funded. A few years ago, I happened to find it. I think it was 4 pages long.
Wow.
And now you have to write
The good old
a
days.
book to turn in an NIH proposal. So there's been a huge change in that. And I know the research part of it was just 3 or 4 paragraphs. And
Wow.
And I talked about what we'd done in Italy with and I was going to do this with fungal spores, but so that's been a huge change. And NIH funded my work and also the bacteriophage work and our giant virus work on the algae for 47 years.
Wow.
And but I don't have funding from them now. And then I've had funding from the National Science Foundation, some. I've had funding from the Department of Energy and even some funding from the Department of Agriculture. I mean, maybe 1 3-year grant. And then if you talk to people about the EPSCoR state, us being an EPSCoR state.
I haven't done that yet, no. You could tell them.
Oh, okay.
If you want.
So I don't know, 25 years ago or so, the Congress came up with where representatives from certain states felt like they were not getting any share of public money funding for research at their university. And so they came up with a group of, I think, around 20 states that are considered EPSCoR states. That is, they receive less money than you might expect them to. And so Congress, I think, sets aside 2% of the budgets of all these agencies like the National Science Foundation, NIH, for EPSCoR states. And I've been involved in at least 2 or 3 of these. And again, these typically go across state lines, and you can hook up with 2 or 3 other EPSCoR states. But in fact, the most recent one I had was with Delaware, Rhode Island, Nebraska, and Hawaii.
So they're $10 million amounts or $20 million. It's a lot of money bringing people together, researchers together at an institution or multiple institutions to focus on one research problem.
Yeah.
And really make progress.
Yeah.
It's amazing how good it is.
Yeah. Well, certainly we're going to talk about the Nebraska Center for Virology.
Well, yeah, you could do that now.
Oh, okay.
Yeah.
So I'm part of the Nebraska Center for Virology, which came out of, and it's gotten EPSCoR money and set up actually by EPSCoR money. I'll get to that in a minute. So probably, I'm guessing now, in about 1985, we had a new faculty member in vet science who studied viruses, who had come from St. Jude's Hospital in Tennessee. And one of his colleagues had gone to the medical school at the same time from St. Jude's, and both of them studied viruses. And so at the time, us people on the UNL campus knew that we had people studying viruses across the campus, and we knew people were studying obviously in the med center, but we really didn't know each other or talked about what we did. So these two new faculty members said, gee, we ought to have a meeting of just one afternoon, and people from both campuses come and talk briefly about what they do, introduce yourself. So we did that, and at a park between Omaha and Lincoln, and it was I think a late September, early October day, and the building we were in had been locked up, and it had more flies. It had to be a hot day, and there were flies everywhere. So we spent half the day grabbing flies, and so this meeting subsequently got called the Fly Swat meeting after that. Well, anyhow, we thought that went pretty well, so we decided we'll do it every year, and it became where we would meet and actually stay overnight in the park and have almost a 2-day meeting, and people would bring grad students, and so we'd get up to 100 people because we now expanded to the other campuses, and anybody even working in a commercial operation that was interested in viruses could come. So in about 2000 or so, back to this EPSCoR program, the National Institutes of Health, which really didn't like the program, but was forced to follow it, announced that every state could turn in one proposal, the EPSCoR proposal, and so since we had the virologists who had somewhat all been working together, we put a proposal in, and it was selected by UNL to be the proposal that went in, and we were successful. And
what year was this?
10 points, probably about 2003, somewhere in there. Okay. And we were successful and got $10.5 million from NIH to set up this. And at that point, then this included also people from Creighton Medical School. And so we were able to hire 3 or 4 new faculty members, and most of these were in life sciences department, School of Life Sciences. And so that built up. Now, at the end of that 5 years, it was supposed to be a one-time thing, but they said, well, some schools or states are doing a good job. We'll let you try again. So we put out another one, the same one, expanded it, and got another $10.5 million. And so during that time, there was interest in the UNL, mainly by Prim Paul, who was a former director of the Office of Research, to maybe build a building and put all the virologists on the UNL campus in one building. And so there was also a program at that time by NIH that you could ask for a building. And after our 3rd try, we had the highest rated proposal at NIH. But that year, because of funding, they wiped out the whole program.
Oh, man.
So anyhow, people in the foundation and Prim Paulnet talked this fellow, Ken Morrison, who I'd had some contact with earlier, into donating money, for which we do have a building called Morrison Hall now, where all the virologists on UNL campus are located. So there's an interesting side story to that, in that after the building was built and we were moved in, the when Obama became president, there was this interest in stimulating the economy in that. And so NIH remembered our proposal and said, called up somebody, I assume Prim Paul, said, could you use $7 million? And that takes like a microsecond to say yes. So we actually have an addition on the building that was built from NIH money. And it was supposed to be for hiring 6 more virologists, but and we had some money to do that. But this was a time when Charles Wood, who was really the director of the center, and myself as assistant, made a mistake in that when we were getting the money like every year to hire another virologist, we decided to pool it because we knew we wanted to find a new director. And so then when one of the budget crises came along, probably 7, 8 years ago now, they saw this pot of money that we were saving for the new director to hire people. They took it all away from us. So the addition got built, and now we have people from engineering in that wing. So anyhow, the center for the most part has been quite successful. Charles Wood moved on to Louisiana State University Medical School, but now Eric Weaver is our current director, and a fellow by the name of Hernan Garcia is also his assistant director of the center.
You told us a little bit about the techniques and instruments that you use, but what else? What other methods?
Okay, well, we certainly use a lot of ultracentrifugation to purify viruses, and sucrose density gradients, which are used with an ultracentrifuge to isolate and purify, which was a technique, as I mentioned earlier, developed by Myron Brackey. And so the rest of our lab is pretty much equipment that you would use in a standard molecular biology lab. We do a lot of electrophoresis and both for protein isolation and nucleic acid isolation. And so we do a lot of cloning, which again, one would expect in a lab like us, cloning various virus genes and studying them individually. So trying to think if there's anything else.
So you study the proteins that are expressed?
Yeah.
And try to figure out their activities?
Yeah. So to put that into perspective, what Mark is asking is, so the viruses that you're most familiar with, like flu virus, COVID virus, HIV virus, they have 10 or 12 genes, or 13. Ours have as many as 450 genes, and now there's ones that have been found that are even much bigger than ours. In fact, some viruses now you can even see with a light microscope. So the definition of virology is sort of having to change with it. Although even many of the people that study flu and that have they've probably heard of giant viruses, but don't know anything about them. In fact, the biggest one now has over 2,000 genes, which is more genes than many bacteria. In fact, ours have more genes than some bacteria, and even some eukaryotic organisms don't have as many as that. It's interesting. The fellow that discovered the biggest one, I actually had an email from him this morning because there's a meeting every 2 years on giant viruses it's next month in Germany in a castle south of Munich. And the castle is owned by the Max Planck Society, which is a scientific organization in Germany. And that's held just for meetings. The only problem is it's nice to stay in a castle. It's not something you get to do every day. And the one problem is it's limited to about 70 people because it kept fitting more people in the meeting room than that. So, yeah, that's coming up in about 3 weeks, 4 weeks.
All right. So was your research ever scooped, or did you and another group tie in publishing similar results at the same time?
Well, I don't know if I've been scooped in the sense. I mean, we've always been or I've always felt like even from the beginning, we gave out our virus and hosts to anybody that requested it. And the only time I've been sort of disgusted with a group in Japan that we gave it to, they they tried to publish a paper showing the same thing we'd shown 2 years earlier, and I happened to be fortunate enough to get it as a reviewer, and was not too happy with that group. I mean, I've known them since, and they've now retired, but that was, and I know they knew that we'd published the paper because we'd given them everything.
But they hadn't cited your paper.
No, they hadn't cited us, so, but that isn't quite what you were asking me.
But it is similar.
But no, we've been very fortunate. I mean, we've got collaborators that we work with from all over on various aspects of these viruses, and they've been sometimes overly generous. In fact, back to that potassium ion channel protein, I said there's 60 publications on it. I told most of them by this couple from France and Germany, and I told them years ago you don't have to put my name on all of them, but they still put it on. Out of 60, I'm probably on 30 of them.
Oh, I see.
But they've done so many things with this channel that some of it I don't even understand in terms of how these channels work. By the way, that's a very important problem in medical science. Every human has probably 100 different potassium channels in their body, and they all have a common core region, and our virus just encodes that core region.
Yeah, so this question is really about competition and cooperation, as you're suggesting. People working in the same area. And then how does that play into your research, right? So then, were your published results ever contradicted, and how was it resolved?
No, I mean, we've been very fortunate that so far we've not had any reason and any people saying that, gee, you're totally wrong. So, I mean, there's a couple papers I've published that, in retrospect, probably I wish we hadn't published, but in general, we've never had anybody say well, there's one question. So one thing we haven't talked about is that at least one of these viruses we've discovered may be involved or associated with certain mental health issues. And this started, and I'll get back to where that's to direct answer your question. So this started about, probably 15 years ago now, when one afternoon I got a phone call from a group at Johns Hopkins University Medical School. And they studied schizophrenia and bipolar diseases. And they've always been convinced that besides probably a genetic component, there's probably an environmental or viral association with these diseases. So they have lots of stocks of people who had died, brain tissue, from people who died from these diseases. And they had done a large, what they call transcription analysis. That is, they had isolated RNA from a lot of these brain tissues and sequenced it, see what was there.
But you could find out what actually is going on in those cells.
Yeah, that's what but they were also looking for foreign things, viruses in this case. And so they got hits to, this is what they told me. So we got a bunch of hits to chloroviruses. I said, what are you talking about? And then they told me this. And they said, would you want to pursue this? I said, well, it sounds interesting. And 2 days later, I had 2 people in my lab. That's how serious they were about it. So anyhow, we published 3 or 4 or 5 papers with them on various aspects of that. And then the money they had dried up. That we'd gotten some money from them. But during that time, at a social, I met an MD in Lincoln who studies Lou Gehrig's disease. He's probably the one person in southeastern Nebraska that patients have Lou Gehrig's disease go see. And he said that this is one of these diseases. We don't know what causes it. A lot of people, I mean, not a lot, but people come down with it, and it's a death sentence. I mean, that's when you're going to die from. And he said, maybe your virus has something to do with that. No, I can't. Impossible. They said, well, what if I get you a little bit of money? Would you look at some samples I could get you? And this is one of these projects that 7, 8 years later still doesn't go away. There seems to be an association with one of the viruses that we've tested. We've only tested 3 or 4 of them with Lou Gehrig's disease, or people who have Lou Gehrig's disease. In fact, the latest set of experiments, and we've been able to get some foundation money now to pursue this, mainly through this MD in Lincoln named Gary Petit. But the latest set of experiments, and we do this work with Tom Petro in the dental school here, who's a molecular biologist, and was where we've looked to see if patients with Lou Gehrig's disease, and we've gotten brain tissue now, or blood samples from one of these foundations that have a bunch of, and look to see if they have antibody to this one virus. And it's clear that the patients that died from Lou Gehrig's disease compared to controls have antibodies to that one particular virus.
Wow.
And we've also looked at cerebral neural, what's that, CNS? You probably know more animal stuff than I do. And that has the same results. So obviously, Gary Petit's very excited about this. The other thing Tom's done a few years ago, we took certain animal cells and tried to infect with the different viruses. And this one particular virus looked like it replicated in the host, human host cell, by at least statistically, it said it was significant. And it also induced many of these cytokines, and these are things I don't know much about, like IL-6 and that with the known to exist with the disease.
Yeah.
So there's now probably 7 or 8, maybe 10 papers in total between the two, what started with Johns Hopkins and this more recent work here. So, okay. So then back to the question I have everybody challenged.
Yeah.
So we published the first paper from Johns Hopkins, I think in PNA, preceded the National Academy of Science, and a group in either somewhere in Norway a few years later challenged that one in saying that they had looked at some of the reagents, and they could find a little bit of evidence of our virus sequence in those. But we replied, primarily the group at Johns Hopkins said that we did enough controls and had 3 or 4 arguments why they didn't think that's the case. So that's the one case where there was at least something. As far as I know, there hasn't been anything challenging us since on that.
But when you've got a virus that's ubiquitous and you can't kill viruses very well, it's really hard to get them out of your systems.
Well, yeah, even though as far as we know,
they're falling all over the place, right?
Replicate.
They're everywhere.
It's something that needs light to the host to grow in.
Yeah.
Now, as I say, now we've found them in maybe replicated in some animal cells.
Mhm.
The other story I did, or the other aspect of this I didn't mention, was one of the experiments we did with Johns Hopkins was I would send them the virus, and I told them and send them the host, and I said infect the host, and then 4 hours later feed it to the mice. And we had, and the reason I did that is one of the fascinating things is these viruses encode for a gene to make hyaluronic acid, which had never been found outside of human, well, vertebrates and a few plant or pathogenic bacteria. So, and I thought, well, maybe that's one way they could get in. So anyhow, then we had the group at Hopkins cut out various organs at 1, 2, and 4 weeks, and then they sent them to us, and then we tried to assay for plaque assay for infectious virus. And after 4 weeks, 3 out of 4 mice were in the spleen, and this was all done blindly. It was only when we got finished that we talked to them, we knew which hundreds of samples, which were which. Actually had infectious virus in it 4 weeks after we had fed them the virus, which suggests probably there's some form of maybe replication somewhere going on. Or if it was the original virus, to think it survived that long would be, I would think, quite unusual. So, yeah.
How does stuff get into the spleen? I'm going to go.
What's that?
I'm going to go off topic here. How do things get into the spleen? Is it from the liver that it flows to the spleen?
That's where I see my animal background is zero.
So anyway,
but people tell me that that might be a place where they would be. So
All right.
I wish I knew more. I should know.
So we're going to step away then from research and go on to career activities. Again, being a research professor at a research university is a complicated thing. How important were societies to your career? Which conferences do you regularly attend? And was there a meeting or meetings that were especially memorable?
Well, I've gone to a variety of meetings over the years. I mean, initially when I started, since I was in a plant pathology department, I went to the annual plant pathology, what's called phytopathology meeting for 3 or 4 years. I've been to the American Society of Microbiology meetings, and then probably and been invited to give talks at both of them. So, I mean, major talks. And finally, in about probably 1983, I went to the International Virology meeting in Manitoba, Canada, because I said, well, gee, maybe I'll become a virologist. By the way, I've never had a course in virology in that life. So it's one of these things where you never know what you might end up doing. So I have huge deficiencies in knowing a lot generally about viruses. Of course, I feel pretty comfortable talking about giant viruses. But anyhow, so after that, then I started going to the American Society of Virology meeting about every year. Actually, I've been invited to give 3 major talks there, including one year the keynote address, which was quite surprising because usually they'd have people that talked on HIV or flu or something. But so I feel pretty honored in doing that. Then over the years, we started viruses on algae, a group in Norway did, in about 2000, somewhere there. And I was invited to that. That was interesting. Another, have to tell stories, you get old. So I flew to Newark, New Jersey, then to get a flight to Oslo. And they told us in Newark that the flight controllers in Norway may go on strike. So anyhow, our plane ended up in Sweden, and they had buses there to take us to Oslo. So, but I happened to get on the one bus where the driver didn't know where Oslo was, and it turns out about an hour after we're driving on the highway, he stops and flags somebody down, and they said that way, backward. So an hour later, we're going back by the same airport, and we got into Oslo about 9 o'clock on a Saturday night on the meet, and the meeting was in Bergen, which is the other side of Norway.
Oh,
So I had met a couple people on the bus who lived in Bergen, had nothing to do with the meeting.
mm,
And so we go to the train station. And of course, all the trains were totally full because they knew this was going to happen.
mm,
So and all the rental car places were closed.
mm.
And so I just said, well, what about a taxi? And I had no idea where Bergen was. And they looked at me sort of funny. And then about that time, Russ Meitz, who was the co-discoverer of the algal viruses with me, the guy I was drinking beer with way back
Mhm.
before, shows up because it ended up in Denmark and
Oh.
somehow taken a ferry
Oh.
or something across to Oslo. And so now there's four of us. Well, anyhow, the guy looked at me and I said, taxi. And he said, well, I'll go call. So this woman shows up in a brand new Mercedes,
Ooh.
and the four of us pile in. And then that day, I mean, it was in June, so there was about 20 minutes of darkness. We drove to Bergen and wouldn't stop once. And turns out she raced motorcycles on a weekend because we were going like crazy through the mountains. And so we get into Bergen at 3 in the morning or 4 in the morning. Had to wake up people at the hotel, and then I let the two guys from Bergen negotiate how much this is going to cost. So they settled on $200 a person, which probably wasn't too bad because you would have stayed in a hotel in Oslo. Well, yeah, the next morning or next afternoon, I finally met the people who were hosting the meeting. They said, "How'd you get here?" I said, "By cab." And their face fell thinking, we got to pay for this.
Yeah.
And I told them what it was and how it worked out. But the thing that they couldn't believe, and everybody I talked to, and I said, we took us, I'm making up the number now, 4.5 hours. And they said, that's impossible to drive from Oslo to Bergen.
That's an 8-hour drive.
Nobody can do it. It's possible. And they must have, everybody I met from Norway said the same thing. So anyhow, we postponed the meeting for a day, and people were coming in all over from different ways of getting there because the airlines were closed. But anyhow, that was the first of the aquatic virus meetings, and we've now, I think, are on, I don't know, 8 or 9. The latest one was in France this year. I didn't go to that one. But then back to the giant virus meeting. So a fellow from works for one of the Max Planck Institutes set up this meeting. He was actually got his PhD in Vancouver, and I was on his PhD committee by phone or Zoom, I guess, at that time. Anyhow, he's gotten money to set this giant virus meeting up, and we've gone every 2 years except for the COVID year. We canceled it, and this will be the 6th one. And I've been fortunate enough to be at all of them, and that's always a great meeting because it's people who are working on these new viruses which they're discovering right and left, and some of them really are just don't even look like viruses. They're really interesting organisms, let's say. But the other part of the meeting thing was, so probably back in the mid-1980s, after we had discovered our virus, had some EM pictures and knew it had a giant big DNA genome. I was trying to find out who we can talk to because there's a lot of viruses that infect plants, but they're all little tiny RNA viruses and so on. And the one virus group that it was the pox viruses, which had double-stranded DNA genomes. Turns out we now know they're evolutionary relatives of our smallpox viruses. So, but there was a visitor at Nebraska, and I don't even know who brought him in, by the name of Bernie Moss, who was at NIH, who was a Mr. Pox virus. And so I happened to hear he was in town and just went and met him. And he said, well, why don't you come to the pox virus meeting? which that year was in Heidelberg. And I said, oh, sounds fun. So I put in an abstract called "The Chloral Virus is Looking for a Home." And went and gave a talk and met a lot of the people in that arena. And of course, most of them are working on the disease aspect and so forth. But anyhow, that was a fun meeting. I was always glad I went to that.
Mhm.
And I went to it 3 or 4 times before we started having our own group of meetings.
Mhm. Next would be, did you take sabbaticals? What did you do? And did it ever change your direction?
Okay, I never took a formal sabbatical, but I 1 year, probably 25 years ago, well, maybe even 30 years ago, I spent a month in Australia. One of my former postdocs from Australia had gone back to Australia and was in Canberra at the University of Australian National University or was, and he invited me to come down for a month, and we put in wrote a 1 day proposal to NSF, and it was a lousy proposal, but I called up, I don't know, 6 months later and said, you got money to go to Australia. So I did spend a month down there, but it did not change my research. I mean, I was working on what I was doing here.
Right.
Wrote a review while I was there.
Okay.
Mhm.
So service to your department, to the college, to the university, and I know you've done all kinds of chairing major um, new thrusts. So tell me about the service contributions that you remember the most.
Well, as far as UNL is concerned, I've been head of a number of search committees. I was the head of the search committee when we hired Chancellor Harvey Perlman. I was on the search committee when we hired Ronnie Green, who subsequently became chancellor at the university. I was the head of the search committee that hired Don Weeks as the first director of biotechnology at UNL. I was head of the search committee that hired our Ann Vitterer. I've been fortunate. I've had 4 department heads in 60 years. And the second one was Ann Vitterer, who was a woman that discovered that interesting bacterial virus many years before. And as far as we know, she was the first woman to become chair of a department in a traditional agri school department. I
Wow.
think that's still true. I think that was true.
Mm.
And and of course, I shared a lab with her and did work research with her. And so, yeah, most of my service is at the university has probably been involved with search committees. I've certainly, over the years, turned down many opportunities to apply for department heads, but I've never wanted to do that. And so I've never wanted to be an administrator.
What courses did you teach more often than others? And then what are your thoughts about that?
Well, so my original appointment was 90% teach or research and 10% teaching. So I taught a course in the physiology and biochemistry of fungi for I don't know, 5 or 6, 7 times. It was like every other year, as I remember. And then I also taught. I was asked by the biochem and nutrition department before they became a totally independent biochemistry department to teach nucleic acids in 1 year, because at least I could spell RNA. And I split the course. Somebody was teaching a protein part from their department. I did that a couple times, and then the chemistry department had their own course in nucleic acids taught by a fellow by the name of Nabagupta, who I knew quite well. And I said, this is stupid. We might as well just have one course. So, and he taught most of it, and I gave a few lectures on it a few times, and then dropped out of it totally. And then one of the more fun, well, then I've done 2 other courses. the plant pathology department, we put together an advanced plant pathology course, mainly dealing with biochemistry and fungi, and molecular biology of interaction of organisms and plants. And I was involved in setting that up and did that for a few years. But the most fun thing I think I did was probably starting in maybe year 2010, was with the honors program. The head of the honors program by the name of Patrice Boucher and a guy in the biochem department, Steve Ragsdale, had talked about putting together a course for honors students on creativity in the arts and sciences. And people really traditionally never think of creativity in the sciences and engineering. So anyhow, when they did that, I was asked to participate the first year they had it. And and so you were always paired up with somebody from the social sciences or art. And I was paired up with a fellow from the Quilt Center who was quite a famous person in his own part. Well, anyhow, I thought it was fun. What we did was give like a 20 to a 5-minute lecture on what we did and anything we wanted to say about creativity and so forth, what we did and why we did it.
Mm-hmm.
And so the fellow that had originally started it from Mark Greep, or not Greep,
Ragsdale.
Steve Ragsdale had moved to Michigan. So I called Patrice and said, "This sounds like fun. Would you want to continue it? And I'll be glad to participate, maybe organize the speakers." So we did that for 10 or 12 years. And when we finished up, we'd have as many as 24 speakers, 12 of each. And all these speakers were named professors and some very quite well-known ones. And so that was really fun. We'd do it on a Wednesday night in a classroom. So we didn't have a time on it. It was supposed to last an hour and 15 minutes. But so I'd have each speaker talk for 15, 20 minutes, or well, 20, 25 minutes. Then the students could ask questions, and then the students had to write a reflection paper after each class. But I think I told you before, one of the more memorable cases I'll never forget, I had a fellow from the physics department paired up with Hilda Ross, who was a poet in the English department. And the physics fellow gave his spiel and put up a slide and said, "Here's several Nobel Prize people I know are high-named physicists." And this just made him passing the comment that several people who had worked with this one guy had committed suicide. And Hilda Ross got up and said, "I had a brother who was a physicist who worked for this guy and committed suicide." They didn't even know each other. I remember I almost fell out of my chair, and Patrice and I don't even think all the students totally caught it, but of course they talked afterwards, but I'll never forget that night. The odds of that happening have to be one. You think you could win the national lottery, there'd be better chances. But anyhow, that was a fun class, and we dropped it when the other fellow retired and we did it a couple times after he retired, but then he wanted to move to Seattle to be my nearest children, so that got dropped.
All right, let's see. Anything else you'd like to add about research before we get into your youth, followed by your undergraduate and graduate training and personal?
I can't think of anything.
All right.
Think of it tonight.
Covered a lot of ground here.
Yeah.
Amazing stories. So tell us about your youth. Where did you grow up and what was your childhood like?
Okay, so my father was, so I was born in Virginia, as you mentioned, where my father was a chemist with the U.S. Department of Agriculture, and they had built, and my both my mom and dad were from Central Illinois. They were both grew up on farms. So the USDA Department of Agriculture opened this brand new lab in Peoria in about 1939. And so my dad, he'd gotten his chemistry degree in Illinois in the middle of the Depression, and had been fortunate enough to work in the lab as an undergraduate with a guy by the name of Davignon, who eventually won the Nobel Prize. And so my dad got his degree, but went back to the farm and thought, there was chemists were a dime a dozen during the Depression. And that's one of these stories I almost emotionally break up on. He said one day Davignon had moved to George Washington University in Washington, D.C., and sent him a letter and said, would you like to come and be my technician? And my dad said he got the letter, went behind the barn and cried because he wanted so much to be a chemist.
Right.
And so anyhow, he moved on to Washington and worked for Davignon, and then got a job with the I guess he didn't work for the USDA when he was in DC. So he got a job at Davignon. And then when this new lab opened in Peoria, he applied to be an analytical chemist. And he said out of 190 people they took, they had to take some kind of job or some kind of a test. He apparently got the highest grade. So he moved to Peoria, which I had been born in Virginia, but I was only a year old when we moved to Peoria. And we initially lived right across the street from the lab. We rented a house. And then about that time, penicillin became a big issue because the English had discovered it, obviously, and had chemists working on it. And it purified enough to test one patient who had a high temperature, and the next day the temperature had gone down almost zero. They'd given the world supply of penicillin to this patient. And that's probably about 1939, 1940. Well, the English were getting bombed so much, they brought penicillin to the United States. And so the U.S. had a project just almost like the Manhattan Project, it wasn't as big, but to develop penicillin. And so they had several universities and drug companies. And this new lab in Peoria also was asked to participate. And so there were 12 scientists that were involved in the penicillin project. And my dad was one of them as the analytical chemist. Now, another side story, and I'll show you this painting when we leave.
Good.
I've told you this story, I think.
Yeah.
So they had two people, mycologists out of these 12 that studied fungi or were mycologists, and they were getting in cultures, these green cultures from all over the world to try to find a culture that would produce more penicillin. See, the original culture that was discovered by Fleming just produced minute amount, and they didn't know anything about the chemistry either. It turns out it had both both acid and basic properties, so it was difficult to purify. So anyhow, the secretary for these two people who studied fungi was in downtown Peoria over a lunch break and was looking at this fruit stand and saw a cantaloupe that had some green stuff on it. And she brought it back to the lab, and turns out it produced many more times penicillin than anything they had before. Her name was Mary, and they always called her Moldy Mary after that. And so by the end of the war, they were producing lots of penicillin, saving thousands of lives. And Norm Rockwell painted a picture depicting of this lady looking at the cantaloupe. And when my dad died, that was the one picture he had cut off the Saturday Evening Post or something that I wanted to have with me, and I still have it hanging in my lab. So anyhow, we lived in Peoria for a couple of years, and then moved to East Peoria, which was the wrong side of the river at that time in Peoria. about 3 miles from Caterpillar Tractor Company. And so all my classmates in grade school and high school, for the most part, their parents had moved to Peoria area to work at Caterpillar during World War II, and they came from Arkansas and Tennessee and so forth. And so to put this into perspective, and the culture issue, when I started high school, I think there were 450 in my class. 225 graduated, and 12 or 13 of us went to college. So a lot of them went to work at Caterpillar, which now has left Peoria area, but one time employed 30,000 people, 3 shifts and so forth. So it was by far the primary employer in there. So then going from that to I went to Carleton College, which was a
Well, no,
go ahead.
just so how did you develop an interest in science or math, or then decide to go to Carleton?
Okay, well, I think out of all my friends, both my mom and dad had gone to college, and so I guess I was always encouraged. I guess I always just thought I'd go
assumed you
to
would.
college, and so whereas that wasn't true of most of my classmates. So that was never very much of an issue. And but I went from that environment to Carleton College, which at the time was, well, still is very well known private, small private
I think it's
school.
better known today than it was back in the 60s. So how did you choose Carleton?
Well, actually, okay. So I didn't know where I wanted to go to college. And so my girlfriend and subsequent wife, first wife, she had an uncle that was at Stanford, and nobody knew anything about colleges around where I was going. And she wanted to go to a small college and ask him to list some small colleges that were good to go to. And so Carleton was on the list. And she applied and didn't get in.
Oh, wow.
And she ended up at Lawrence College. But Carleton at that time, I don't remember the Chicago Tribune or somebody came out of this study, and Carleton was ranked third in the country for small liberal arts colleges.
Recently, you mean?
No, that was
back then? Oh,
that was
really?
1956
Oh,
to 57.
I didn't know that.
Yeah.
Okay.
So
They've been at the top for a long time.
Yeah.
Wow.
Yeah, and a lot of money.
Yeah.
I mean, so I mean, I had classmates who were like debutantes.
Oh.
Hell, in my high school, whatever, a debutante. In fact,
know what it was.
I made the comment to one of them that must have been the only girl in my class that wasn't pregnant when she graduated. That didn't go over very well. But and they'd come out of prep schools and stuff, and so I mean, it was quite a challenge. But I certainly owe my whole career to getting back to science. So I didn't really know what I wanted to do in my freshman year. I took chemistry and did okay, and my dad was a chemist, but it really didn't turn me on. So and I'd gotten a D in biology in high school, mainly because I screwed around. And so that was the furthest thing from my mind. So as I worked all 4 summers when I was in college, 1 summer at Caterpillar and 3 summers in a steel mill. Well, anyhow, I was getting ready to go back to college for my sophomore year, and my dad, who said was a chemist and worked on penicillin project over World War II and did other things, said I've got to go to the lab on a Saturday afternoon and shut a column off or something. Said, you want to go? And I said, oh, okay. And he said, oh, there's a guy down the hall I see working. You might introduce you to him. And his name was Wickerham, who studied yeast genetics. And we said, what are you doing? He says, well, I'm looking at some lyophilized cultures. And I said, what's that? He said, well, we freeze dry these yeast cells, and then we can store them and bring them back to life. For some reason, that just intrigued the hell out of me. I said, how do you do this? And I said, how do you study this stuff? And he said, well, you ought to take a course in botany. which is the furthest thing from my
Right.
mind. So I go back to Carleton, and I didn't even know where I thought I knew where the biology department was, and went over and met the chairman of the department and introduced myself. He said, "Well, we've got this brand new guy who just came from Wisconsin who's going to teach botany this fall. I'll take you down and introduce you to him." And his name was Bill Muir. And so anyhow, we hit it off, and I took botany, and that got me excited. We didn't have a course of microbiology in those days at Carleton. Of course, they didn't have one in biochemistry either.
Mhm.
But
Mhm.
so that got me. Then I sort of I took a course of plant physiology and mycology, but I was the only one, so I just looked at the book occasionally and got a fool around in the lab that
Yeah.
he had. And so then I applied to grad school at Illinois in plant path, and my grades were, let's say, average. And but I'm taking chemistry through organic chemistry. I liked organic chemistry.
Mhm.
And so then I applied to 4 or 5 places. And then so in plant path, so I got a couple of scholarships. I went to Illinois, and the guy that offered me the scholarship worked on corn diseases. And it turns out I'm somewhat allergic to corn pollen,
Oh,
so
really?
I didn't know. I mean, I knew I had a fall
Mm.
allergy, but the chairman of the department at Illinois said, "Well, there's this other guy out here that works on antibiotics. He might want to talk to you." So I go out there, and it was one of those cases where I walked in the room and shakers and stuff growing. I immediately knew that's what I wanted to do. So anyhow, I finish out this story, which is interesting. So I he accepted me. So I graduated from Carleton. A week later, get married. A week later, head to Champaign to start grad school. That's Saturday. Sunday morning, I go over and buy a newspaper. Headlines: my advisor had been in a major car crash,
Oh.
killed his wife and 2 kids,
Oh.
and he spent the summer in the hospital. The only reason he lived,
Mm.
he apparently was in the back seat.
Mm.
And
Mm.
and I didn't. I mean, that's
Yeah.
literally my introduction. I hadn't been in town 24 hours,
Mm.
and of course, I didn't know what was going to happen. And anyhow, he spent the summer in the hospital, and he had just hired somebody, a biochemist from Harvard, who originally had a connection to Illinois, to sort of help run the lab. And that fellow, he and I were on the payroll at the same time, but he sort of kept the lab together, which had, I don't know, 4 or 5 grad students and maybe a postdoc or two. So, but then I've got to finish up going back to where I am now,
Mhm.
which you never know how things are going to turn out.
Mhm.
So my advisor had set up a meeting on fungal physiology related to plant pathology to be held at Illinois. They owned a house about 30 miles out that had a place you could hold small meetings at. And of course, he couldn't go to the meeting because he was in the hospital. And so 2 or 3 of those people that he had arranged to come came in on a Saturday morning. Those days we always work Saturdays, by the way. So and if not Sundays. So anyhow, that's but the first day my advisor actually showed up in the lab, and there was this tall guy who smoked like crazy that was talking about what he did. And his name was Mike Daly, who actually was in the plant pathology department, uh, and he moved over to biochemistry.
Mm.
That's a job I eventually took 5 years
Oh,
or 6 years
wow.
later.
Wow.
So, but anyhow, little did I know it at the
No, I
time.
know. Small
Here's a
world.
guy I met the summer of
Super small world.
1960 that,
Yeah.
in 1966, I'd be in his old lab
Yeah.
back in Lincoln. So,
Wow. What a story. Then how did you choose your postdoc research project?
okay, that's so. So my former wife and I had never really been out of the Midwest, even though I mean, to a certain extent, we
Yeah.
traveled a little bit. And so she taught school for the last 4 years. We were at Illinois, and we met several couples through her school teachers, because a lot of them were siblings of people going to grad school. It's pretty standard in Champaign.
Sure.
And
Smart people.
yeah, so we became very good friends with this couple who had spent a year in Austria on some kind of program. So we thought, geez, is there any way we could go to Europe? And so I applied for several different fellowships to Europe, including one to Czechoslovakia, Prague. And that was when they flew us out for an interview. The first time I'd ever been on an airplane in New York. And I think it's probably run by the CIA in retrospect. I didn't know that. But anyhow, I met one or two other of the candidates, and they were in political science or something. But I was the only one who was a scientist. The only reason I'd applied for that was my advisor who knew one of the chemists at some university. Well, yeah, luckily I didn't get that. But I'd applied to the National Science Foundation, and somewhere that fall, as I was applying, this guy from Italy came through the lab, and he'd gotten his master's degree with my advisor several years before. And he said, and I asked, we always had a coffee, and I said, "Oh, what do you do?" And he explained a little bit to me. I said, "Well, gee, we had a journal club every week. We had to give a paper every week." I said, "I just gave a talk on this, something like that." It turned out it was his work.
Oh,
I didn't
wow.
know that because I didn't recognize the name. So anyhow, he said, "Well, why don't you come and work in my lab," which was in Pavia, Italy, which at that time, a lot of the Italian labs weren't that great, but this was in the Department of Genetics there. It was headed by a guy by the name of Luca Cavalli-Sforza, who was a very famous population geneticist. And so that was a very good department. They even had some money from the National Science Foundation. I didn't know any of it. So anyhow, I put his name down for the National Science Foundation, and that was the letter I got a week or two before Nebraska called when I almost didn't come. So that's how I ended up there. And as I say, it turned out to be the real turning point in terms of my research. And it was a wonderful experience. I could start an experiment Saturday morning and make a transfer Sunday night. And you could drive to Switzerland, France, Austria, for the Saturday night and so forth, and all over northern Italy. So it really worked out well. And then I came back and changed what I was going to do here. And with the support of the department, I've been very fortunate.
Sounds great. What research and writing did you do to merit authorship on your very first paper? Do you remember?
Yeah, it's in the journal. So in Illinois, I worked on, my advisor for some reason was interested in aging of fungi. So I developed a way to study this in fungi. And so I had two papers essentially on work I was doing for my master's. And his attitude was, which one do you want to be first author on? And so that was the same way. I had three more on my PhD thing. So it was rather, thinking back, it was rather trivial because things like what you took 6 months to do now you can do in a day. Right. With equipment and so forth.
But collaborations have also made it authorship order more interesting and complicated, and you need more discussions. Back then it was just even though he was I told you the tragedy had happened to him. Yeah.
So actually, that next year, then he spent the whole year traveling
Oh.
with people on work on antibiotics and that worldwide. So I didn't even see him.
Mm.
And so the work was all done in collaboration with this new guy he'd hired, and he wouldn't even let me put his name on the paper.
Oh,
Yeah.
wow.
Yeah, authorship was very different. So I've always been just the opposite. I've probably been very generous on authorship on papers for my lab because it doesn't cost me anything and that might help somebody.
So what prepared you to be able to write a science paper then?
To be what?
To write a science paper, a paper that gets published in a journal. What training did you have in writing?
Well, that's a good question because in English at Carleton, I actually once had an English professor ask me if English was my native language because I was such a bad writer. And so when I was at Illinois, my advisor, I mean, it turns out I was a better writer than most of my classmates because well, all our tests were written tests, and a lot of my fellow classmates in grad school had done nothing but yes or no question or true false or something.
Right.
And but he hooked me up with somebody in the writing department where it was in Illinois, and so I had several lessons with him on writing. I still, I think now, well, I mean, the last 50 years or so, we get almost compliments on our writing, but it's work. I mean, I do have colleagues that, not many, but can almost write a first draft when they write it. And
Yeah.
that's not true of anything I write. It's probably gone over 20 times.
Sure. No, I remember writing the paper, but it didn't seem complicated. You just copied the style of some other paper.
What's that?
When I wrote my first papers, I just copied the style of other papers that had been published and just fill in with all the research detail, right?
Well,
but nobody trained me specifically.
Yeah, that's a good question. I mean, we read a lot of papers because we had to. We didn't give a talk. So, but I don't remember ever quite. I mean, I knew what the style was that the discussion was last, but beyond that, I don't know. At least, or I don't remember how that was. I remember it took me a long time.
All right, so now we're in the last section here, the personal section. Before retirement, what did you do for relaxation or entertainment?
Well, I mean, I played baseball up through college. I one time, like a lot of kids, wanted to be a professional pitcher in baseball, the Cubs, by the way. But I found, well, when I was in high school, a couple of the Peoria area kids who were players in other teams, they get drafted in a Class D, and 2 years later they're back working at Miller. So I didn't think that was going to be a career. Although I did talk to some serious scouts, but but then I always, for some reason, picked up a tennis racket when I was about 8 or 10 years old. Never had a lesson in my life. I still play. I played yesterday, and so, but I don't move much anymore. But I've always liked the game, and so that's probably the one sport. I used to do quite a bit of fishing too, but in the Salt Valley lakes, but I gave that up quite a few years ago.
So tell us about your family. You mentioned a first wife, any kids?
Yeah,
how did that
so
all play into this career?
Yeah, I have my first wife and I were high school sweethearts. We got married in 1960 and then separated in 1975 or so. Well, we had one child who's about 58, a boy who's retired at 48 and lives in Spain. And so he moved with his mother to Ames, Iowa, and grew up in Ames and went to electrical engineering, I think, at Iowa State. But he took a year off at what's it called, co-op or something, where you go work in a company for a year. They did that with some company in early computer days in Minneapolis, and got into, he was one of the first who went to work for Arthur Anderson, which had a separate division that was one of the first, and he was one of the first to learn this ACS program out of Germany for manufacturing. So luckily that then Anderson got in trouble, but but just before they did, Deloitte and Touche came along and said, we'll double or triple your salary and you can live anywhere you want in the United States. So he wanted to live in Hermosa Beach. Well, he wanted to live in California. So he bought a place in California and made enough money there and retired at 48. But part of it was, the travel. I mean, they'd have have jobs where they'd have 150 people where you go to Boeing and totally change their whole computer system to this new system.
Right.
And so and he had a couple of jobs on the East Coast. So, he'd fly out on Sunday afternoon and work all week and fly back Thursday night or something. So it got tiring. And so he had made enough money and got married to a lady who was originally from Spain, and she wanted to go back because her father was ill, and they ended up staying in there. It's been there now 7 or 8 years. I thought he'd get bored after he retired, but is now sort of becoming a gentleman farmer, growing lots of vegetables and stuff. In fact, I'll see him next in a couple of weeks because the week before this giant virus meeting, we're going to Spain to spend a week with him and his wife in southern Spain, which I've never been to. So yeah,
No, never been.
and then so then I was single for about 10 years, and then my current wife came out as a grad student, and we ended up getting married. And of course, I was obviously she worked in my lab, so I was on our PhD committee, but I stepped aside during that. We got married about a year before she actually graduated. And we have 2 girls.
Oh.
One lives in Lincoln with 2 kids, and one in Columbus, Ohio with 5 children.
So then you are still employed. Most people that I'm going to be interviewing are retired. How many years have you been at UNL now?
So this is number 60.
60. How many faculty do you know who've gone longer than 60?
I don't know if I know it. It's not something to go.
Are you trying to get a record here?
But not really. It's a fair question, and obviously the end's coming at some point. But I've been very fortunate. I love what I do. I mean, these viruses have taken us in so many interesting places, and we've been productive. I mean, I've been fortunate in and working with a number of people. I think we've already got 5 or 6 papers out this year. So as long as I'm productive, I'm not going to hang on just to hang on. And so and for the most part, I'm healthy. I do have some old Minn problems, but so at this point, I thought about taking the buyout, which is this year, but I've got 2 people who want to work in my lab next year on sabbatical type things, so I want to be around for that. So we'll see.
All right. Well, with that, thank you very much for talking to me, and I'll see you around.
Okay.